Hello, my name is Layla Taife, and today I am going to demonstrate how we are using CRISPR-Cas mediated phage therapy as an antibacterial agent. Thanks to the International Genetically Engineered Machines Foundation, known as iGEM, we are participating in an annual synthetic biology competition against foreign and domestic teams at an international conference to be held at MIT. The competition criteria includes design of a wiki for the project, concept originality, benefit to society, and proof of concept. Each year, teams identify a clinical and/or societal need and synthesize a biological system to address the issue. Our team is comprised of undergraduates in molecular biology, advised by graduate students, and our PI, Dr. Robin Dowell of the BioFrontiers Institute. For our project, we chose the clinical need for an alternative to antibiotics. Emergence of carbapenem-resistant Enterobacteriaceae has been declared a serious threat to public health by the CDC, while other pan-resistant species, such as tuberculosis and MRSA, still pose imminent risk. Due to the ability of bacteria to share advantageous mutations by conjugation, rapid spread of antibiotic resistance between numerous bacterial species is likely. This is encouraging many companies to abandon antibiotic research in favor of more profitable drugs due to the poor return on investment in lieu of evolved antibiotic resistance. This evolution is causing a resurgence in phage therapy research as a less expensive and more adaptive treatment option. Antibiotics target metabolic pathways shared by many bacterial species and therefore lack the specificity to target only one strain of bacteria. Research into new antibiotics can cost up to $1 billion and take years to complete clinical trials. Conversely, phage infect very specific classes of bacteria based on coat protein recognition and are cheap to produce. Synthetic phage can be engineered to specifically target mutations, such as antibiotic resistance, thereby killing pathogenic strains while leaving endosymbiotic organisms unharmed. CRISPR-Cas is part of the bacterial immune system that targets and destroys foreign DNA. CRISPR RNA directs the Cas protein to the target DNA where Cas cleaves the DNA. We will be using a type 2 synthetic CRISPR array, which makes a single RNA fragment called a synthetic guide RNA, or sgRNA. The sgRNA will guide our Cas9 DNA nuclease enzyme to the target DNA, causing a single double-stranded break in the genomic DNA. This break in genomic DNA will lead to cell death. Infection by a native phage leads to phage multiplication and lysing of the cell. The multiplication of phage carries an inherently high risk of mutations, a major problem of implementing traditional phage therapy in eukaryotic systems. Using synthetic biology, we have engineered a phage with target specificity that lacks the ability to multiply in vivo. By utilizing a helper phage with a weakened packaging signal to build the phage capsid, we can preferentially package a second phagemid lacking the ability to multiply. Thus, by selectively packaging CRISPR-Cas systems into a phage capsid, we can eliminate the possibility of further phage multiplication and target with high specificity. We are currently engineering a spacer design tool that will optimize against off-target effects and measure conservation across a database. This tool will enable users to pick a target organism or organisms for destruction, as well as species to preserve. The program will then recommend spacer sequences. This program, along with the synthesized CRISPR-Cas system, will enable researchers and physicians to counter the evolved resistances to treatment more quickly than bacterial evolution. The phage-delivered CRISPR-Cas system has greater specificity, adaptability, cost efficiency, and fewer adverse effects than antibiotic treatments or current phage therapies. In conclusion, CRISPR-Cas mediated phage therapy is the future of antibacterial medicine.